Well, good afternoon, everyone. I think we'll get started. Our room's almost full, and we've got a lot of speakers with great information to share with you. So I'm Andy Fish. I'm Executive Director of AdvaMedDx. Dr. Amalia Issa from the University of the Sciences is Chair of the Department of Health Policy and Public Health, as well as Director of the Program in Personalized Medicine and Targeted Therapeutics. She's going to give us a perspective from the research side of the diagnostics industry, some of the advances that are going on in research and how they translate into patient care through diagnostics development. Cesar Garcia is President and CEO of IRIS. He's also a member of the Board of Directors of AdvaMedDx, and he's going to share some information about one of their cutting-edge tests in the prostate cancer area and some broader observations as well on the industry and innovation. And Walter Koch, who's Vice President of Head of Global Research at Roche Diagnostics, is going to talk about a new test that they have developed in combination with a melanoma drug. It's a fascinating story, a great example of another advance in the diagnostics field. And last but certainly not least, Marcia, who's President and CEO of the International Cancer Advocacy Network, is going to talk a bit about, from a patient perspective, on how important it is to continue to see innovation in the diagnostics area and the impact that it has on cancer care for patients. Just for a couple minutes before we get to that lineup, I wanted to give a brief introduction of AdvaMedDx and do the world's fastest overview of the field of diagnostics. So AdvaMedDx was founded in 2010 as a division of AdvaMed, which is the Medical Device Trade Association. DX stands for diagnostics. So we are the diagnostics division. We currently represent about 65 member companies from everything from the global industry leaders to early stage startup companies. Our goals are to educate stakeholders and advocate for public policies that advance the use of diagnostics for better patient care. And if you leave here with almost nothing else, take away the fact that diagnostics are an incredibly efficient lever of healthcare value. Currently, Medicare or diagnostics spending accounts for about 2% of Medicare spend, but influence about 70% of healthcare decisions. And I think as you'll understand better after these presentations today, you'll see that that value proposition is just getting stronger every day with the kind of advances that are coming out, particularly in the molecular diagnostics area, and the impact that they're having on patient care. So just briefly, what exactly are we talking about? We're talking about diagnostic tests that are run on samples taken from the body, everything from blood and urine and other fluids to tissue samples. And these tests run the gamut of consumer-friendly, use-at-home tests, like a blood glucose monitor that diabetics use on a daily basis, multiple times a day, to monitor their blood glucose levels, and pregnancy tests. I don't think this one probably needs a pregnancy test to tell her she's pregnant, but it drives the point home. At the other end of the spectrum, we have our companies, some of our larger companies in particular, make highly automated, complex laboratory systems that can perform dozens, even hundreds of different kinds of tests on one sample run through these automated machines, up to a couple hundred samples an hour. Truly amazing what can be done at the high end of the automation spectrum. You can't read this, but I put this up here to promote our diagnostics policy primer, which is available on our website. This is the next couple of charts are taken from that document. This chart indicates the fact that diagnostics are used at every stage of the healthcare continuum, all the way from screening for asymptomatic diseases to early disease detection, to diagnosis itself, to staging disease, prognosis. What stage of disease does this patient have? What's their prognosis going forward? To drug selection or other identification of appropriate therapy, monitoring treatment, to follow-up care. So at every stage of the healthcare continuum, diagnostics are playing a critical role. And diagnostics span a wide range of technologies and disease areas. Some of the different areas are in the chemistry area. That would be, for example, urinalysis or immunochemistry, hematology, the study of various aspects of blood, or cytology, which is the study of cells, microbiology, infectious disease areas. There's a whole range of testing and some examples here up on the slide. To molecular diagnostics, which is an area that's capturing a lot of attention these days, especially in the genetic testing area. Something that some of our speakers will address as well. So that's just a quick overview of the spectrum of diagnostics, technologies and applications across a number of disease areas. And again, as I said, that's in a document on our website. And all these advances are allowing doctors to usually do a little bit better than this. A couple of quick resources for you. Lab Tests Online is a great website. It provides information on the kinds of tests that are used across a wide range of diseases and conditions. It also gives background on the diseases and conditions themselves and really does a great job of explaining the connection between disease states and the kind of diagnostics that clinicians will use to help patients. And finally, our website also is a resource that we continue to build. We have a number of materials on here, including the policy primer that I referenced before. Please take a look. I think you'll find some interesting and useful information there, and links to a number of other organizations as well that are involved in diagnostics. So without any further ado, let's turn to our speakers. Dr. Issa will be first, and welcome Dr. Issa to the podium.
Thank you. So hello, everyone. Thank you, Andy and AdvaMedDx, for inviting me. I'm delighted to be here this afternoon. I'd like to start with a very brief story. A woman is driving here in Washington, D.C., and realizes that she's lost. She pulls over to the side and sees a man sitting at a bus stop, and she says, "Excuse me, sir, can you please help me? I was supposed to meet a friend about an hour ago, but I'm completely lost. Can you help me out? Can you tell me where I am?" And he says, "Sure, I'd be happy to help. You are at 38 degrees longitude and 77 degrees latitude, and you're driving a black BMW." She looks at him quite perplexed and says, "Are you a researcher?" He's surprised and says, "Yes, I am. How did you know?" And she says, "Well, what you've told me is technically accurate, but it's absolutely of no use to me. I'm still lost, and if anything, I'm even more late than I was before." And he looks back at her and he says, "Are you a policymaker?" She says, "Yes, how did you know?" And he says, "Well, you have no idea where you are or where you're going. You made a promise that you can't keep, and you're still exactly where you were before, but now you're blaming me for it." But in all seriousness, it's wonderful to be here because over the past several years, researchers and policymakers have actually found ways to work very well together and have some very fruitful relationships and exchange. So I'm really delighted to be here today. So what I hope to do in the short time that I have with you this afternoon is to explore these two questions. What do we know about the adoption of molecular diagnostics in clinical practice, and what needs to be done for molecular diagnostics to be more routinely adopted into clinical practice? And to show you why molecular diagnostics, and particularly molecular diagnostics as they are being used in cancer, because that's the theme for us this afternoon, are making all the difference in the world for patients right now. So molecular diagnostics are, as Andy said, are tests that identify molecular and genetic markers for an individual patient. They can determine potential benefit from a specific therapy, or they can determine risk of developing a specific disease or health condition, and they're also used for various other purposes as well. So let me introduce you to Betty. Betty was diagnosed with breast cancer some 20 years ago in 1992. Now, what happened when she was diagnosed is that she first received a mastectomy. Then she underwent chemotherapy and then radiotherapy. And all of this, what we now might think of as a shotgun approach to treatment, was at the time standard of care for breast cancer. Now, this is Evelyn. Evelyn was diagnosed with breast cancer about a year ago. Unlike Betty, who was diagnosed 20 years ago, Evelyn had some options. She might have had her tumor profiled to better understand what drugs might work best for the particular type of tumor she had. Her tumor was profiled using one of the newer gene expression profiling tests to understand how aggressive her cancer is and whether it is likely to recur or not. So Evelyn will have had a chance to be treated the right way the first time, right from the get-go. The options are there for her. And this is increasingly becoming current practice. And it is also the future of personalized medicine. And these different components are coming together in a way now that is leading us to ask some fundamental questions about how cancer is characterized and treated. What do we need to know? When do we need to know it? And what difference does it make? What do we need to know specifically when someone is diagnosed with a particular disease or cancer? When do we need to know it? What difference does it make? When do we need to know that you have a disease like breast cancer or colon cancer? What does that mean for you as an individual? And what does it mean for your treatment? And so we're beginning to move towards this new concept, a new era, if you will, of personalized certainty, knowing with a greater degree of accuracy that was not available to us before, a degree of precision that's more in line with what patients and their families and their healthcare providers want. that's appropriate for these types of life and death decisions, providing us with more right treatments for specific subpopulations and individuals than ever before. And this is driving new value propositions that have been unimaginable until now. And this creation of value is what is driving the field of personalized medicine forward. Well, all of us are different. We all know this. And you all know that what's behind our differences in eye or hair color or height or what have you is largely DNA. The 3 billion pieces of data in every cell of your body that is the blueprint for all sorts of things that make you who you are. Whether you are tall or short, whether you have blonde or brown hair, and also how you might get certain diseases or not, and how well you might respond to particular treatments or not. So given that we all understand this and agree that our DNA is largely responsible for our differences, why is it that up to fairly recent times we have been content with a one-size-fits-all approach to therapeutics and disease treatment? Because the fundamental reality is that most diseases are considerably more complex than the size of your feet and whether your shoes fit or not. So you would think that before we can truly understand diseases, particularly something like the many cancers that befall us, and we are all, in one way or another, affected by cancer, because if we don't get it ourselves sooner or later, we have a friend or a loved one who does. We ought to have a good idea of what's going on and what goes wrong at the level of complexity that's appropriate to the disease, and that is often at the molecular level. So when you hear the term diagnosis, if you're like most people, you tend to think of it in somewhat of a reductionist way, quite simply as sort of a binary concept. I am healthy, I'm not healthy. I have cancer, I don't have cancer. I feel great, I don't feel so good. But over the past several years, we've had tremendous advances in the field of molecular biology and in genetics and genomics. We now have technologies such as DNA and whole genome sequencing, polymerase chain reaction, PCR, protein analysis, and proteomics. And all of these mean that we now understand disease at a deeper, more fundamental level than has ever been possible before. So we're beginning to understand at a much deeper molecular level what really makes us sick. And now the really interesting thing is that these research tools, which you see here, which for many people, like many of you in this room, are sort of abstract because they don't necessarily see them. You typically think of them as these cool toys that scientists play with in their research labs, are now increasingly being transferred and translated from the research labs to the clinic. So instead of simply being able to understand what makes a disease, we are now able to apply these types of powerful, sophisticated technologies and tools and understand what makes your disease a disease. And this ability to analyze and characterize individual patient genomes is making all the difference to how we treat your disease. What choices are being made among an increasing array of treatments to improve outcomes? So, going back to my earlier point, you can see that the term diagnosis, in the way that most of us think of it, really is quite a reductionist term, because it doesn't capture the complexity of disease, and it doesn't capture the fundamental interdependence between all the genes and proteins and other elements that make us sick. So, molecular diagnostics are giving us a new ability to characterize disease. And this is a growing area of research, and it's really changing how we think about where the field of personalized medicine is going to be effective. So let's go back to Betty again. When she was diagnosed about 20 years ago, she had to go through a mastectomy with all the trauma and the difficulties associated with that. Then she had to do a round of, perhaps maybe several rounds of chemotherapy. And then she underwent the radiotherapy. And the interesting thing is that we now know that only about maybe 20-25% of women are going to respond to chemotherapy depending upon the type of cancer they have. But we had no idea, especially back then, who the responders were going to be. So everybody got chemotherapy as a matter of course, as a standard of care. And as patients and as a society, we simply had to live with the consequences of this shotgun approach with the side effects of chemotherapy and the costs. But let's take a closer and deeper look at breast cancer. This is what I, as a scientist, and my colleagues think of when we think about breast cancer. This shows what are called signal transduction pathways. So this shows a whole group of proteins and genes, and these are the molecules that convey the messages inside the cells and also between the cells, and there's a lot of crosstalk going on between them. Someone once described this kind of diagram as an electrical wiring diagram gone horribly wrong. But when you see this, you can begin to think of cancer, and this applies really to any cancer, not just breast cancer, but you can begin to think of it as a very complex, incredibly interdependent wiring diagram, perhaps the most complex wiring diagram you might have ever seen in your life. So this is the sort of diagram that we use to describe signal transduction pathways, and this is actually only one of the pathways that may be relevant to breast cancer. So we are increasingly understanding how changes and defects in these molecules drive particular diseases. And equally important, what we are also learning is that breast cancer and many other cancers, it's not just one disease. There are a number of different subtypes of breast cancer. And this is true, by the way, as I said, for all kinds of cancers. There are often many subtypes of those. And so, in fact, scientists and physicians now tend to describe cancers by their molecular structure rather than by their location. So it makes more sense nowadays to refer to KRAS as a cancer, for example, than to simply talk about breast cancer or prostate cancer or lung cancer. Because that makes more sense to describe what's gone wrong, and it starts to lead us in the direction of where we're going to apply the therapy or the treatment. So if we understand at a molecular level where the fault may lie, then that will direct us to where to apply the therapy, the treatment. And so understanding these mechanisms of action and being able to make deeper diagnoses at a molecular level is an absolutely fundamentally, critically important area of research. And when it comes to cancer care, the numbers are staggering. Tens of billions of dollars are spent each year on treating cancer, in this country as well as around the world. Now, if you're still thinking in terms of a one-size-fits-all approach, then first of all, you're not helping the patient, and secondly, you're not doing the healthcare system any good. And we know from studies that have been done that billions of dollars are wasted in the US and in other countries every year on treatments that make no difference to how cancers are being treated. So personalized medicine and the use of molecular diagnostics can not only benefit patients, but also are poised to realize some significant efficiencies in the healthcare system. So let's go back to Evelyn. One of the first things that happened when Evelyn was diagnosed with breast cancer last year is that she had her tumor profiled using molecular diagnostics. That profile turned out to be ER positive, PR HER2 positive, and that would have been immediately and completely relevant to the first line of treatment that she received. And then she might have had her tumor further characterized using gene expression profiling tests, such as Oncotype DX or MammaPrint, which would indicate the likelihood of cancer recurrence. And these tests would have given her a recurrence score. And so within a matter of days, she and her healthcare provider, her physician, would have a better idea of whether she was going to need adjuvant chemotherapy following surgical resection of her tumor or not. So this type of information is valuable for Evelyn and other patients like her in that it can make all the difference in the course and progression of her disease. It can make a difference as to whether Evelyn undergoes a taxing course of chemotherapy treatment, whether she and her family incur the expense of chemotherapy, and whether she loses her hair or not, and is subject to sometimes unnecessary toxic side effects. So as we look to the future for Evelyn and other patients, they are likely to have options, choices in treatments that will make a difference to the right, in terms of getting the right treatment to the right patient at the right time, personalized treatment for their cancer. And this is the fundamental value proposition of molecular diagnostics, providing these options. And this is also making a difference for the pharmaceutical industry and for drug development. If you think about how we're beginning, how clinical trials are being conducted, we're beginning now to increasingly see the stratification by subpopulations of patients in clinical trials. And this is leading to the repurposing of drugs that might not have worked as well before, but now we can repurpose them for another use, relabeling of drugs by the FDA. And over the past year or so, we've been seeing some significant launches of drugs for a number of cancers like melanoma and lung cancer, and you'll hear a little bit more about that shortly, and also prostate cancer. And all of these really are beginning to take advantage of the power of personalized medicine. So we are beginning to use these tools, these biomarkers, to select which patients to include in clinical trials and in drug development, and we are beginning to get drugs to the market a little bit more efficiently. Well, this is all very well as long as you're using molecular tests to rule in medicines or drugs. But what happens when you think about ruling drugs out? Now we have to start thinking about some other factors as part of this equation. And this becomes incredibly important because if you're going to use these technologies, these molecular diagnostics, these tests to determine which therapy should be given, and importantly, what therapies should not be given, ruling drugs out, then you better be sure that you're making the right call. And particularly in the case of cancer, where we're talking about life or death decisions, you want to make sure you're making the right call. And this is where the complexity of decision-making comes into play. So you will want to make sure that the test is validated. And we have all sorts of sophisticated tools from epidemiology and from clinical trials to help with that validation so that you have the confidence to make that call and be able to say, this treatment will not do this patient any good. And you're also going to want to make sure that the test is regulated, and that's what the FDA is for. You'll also want to be sure that the provision of this particular test is commensurate with the degree of accuracy and precision that is required and expected in healthcare in our society today. And you'll want to make sure that these tests and the consequences of their application are going to be interpreted correctly for patients in order to be able for them to make good and sound decisions. So as we are increasingly crossing this translational divide from research to real-world practice and implementation in clinical settings, what we're finding is that all of this is leading to a whole set of new questions. Questions that are making us, as a society, feel as if we're sailing in as yet to be charted waters. Questions related to analytical validity, and clinical utility, whether the right clinical needs are being addressed and being met, whether the applications are acceptable to patients and providers. And these larger health system level issues are exactly the types of questions that we at the program in personalized medicine and targeted therapeutics work on. Our research agenda for some years now has been largely driven by the question, what is needed for personalized medicine to be translated and well integrated into healthcare delivery and health systems? And so one line of investigation, just as an example that we are very active in, has to do with decision making and how patients and providers are using these new molecular diagnostics to make better decisions about treatments, and we have developed instruments to quantify, to measure how patients make decisions about using and adopting molecular genomic diagnostics, and how they make trade-offs between the options that are presented to them. Because as I hope that I have convinced you, as we increasingly use these sophisticated tests, we also increase the complexity of decision-making in clinical settings. So why is this important to investigate? Because for the longest time, since the completion of the Human Genome Project, we've been accumulating lots and lots of data, about 3 billion data points that make up our human genome. But now we've reached the tipping point where we're actually accumulating more comprehension to understand what that data means for us. And with that increased understanding comes the complexity of decision-making. Because if you think about it, if I've convinced you that by now that cancers are complex and that they need complex interpretation, what would that change for you? What would you do with the information that you get about your disease? How might that change your behavior? Because if there's anything that's more complex than signal transduction pathways, it's actually human behavior. This is an artist's rendition of what Michelangelo's famous statue of David might look like with a higher BMI. So, just to give you a very quick example of some of the work we do, this is from a recent paper in which we modeled some of the decisions taken in breast cancer treatment and compared two commercially available gene expression profiling assays, Oncotype DX and MammaPrint, including quality of life and cost-effectiveness issues, and we found an unexpected result. We found that Oncotype DX, which is the most commonly used gene expression profiling test for breast cancer in the US, is actually the test that's less cost-effective, and also it exhibited slightly lower quality-adjusted life years, QALYs. But what's interesting about this is not so much just the results, that is in and of itself interesting, but the fact that policy decisions by payers and others had been made in the absence of this type of evidence. So this was really the first study of this type. And so there's a real need for more translational research studies of this type to be conducted alongside the molecular biology research as well, as we, as a society, continue to implement novel genomic molecular diagnostics into practice. So I've told you about what molecular diagnostics can do and how they are being used for risk prediction, screening, diagnosis, and therapeutics. And I've talked about the value proposition. But the speed with which and how well we manage to adopt new molecular diagnostics into practice hinges on several factors. Certainly decreasing costs, which we're beginning to see in terms of sequencing and technologies. The sequencing instruments are now clinically affordable. Increasing the speed of sequencing, which now we are able to do it within a 10 to 36-hour window. Bioinformatics. This is still very much a roadblock because we still need to be able to figure out better mechanisms, both in terms of bioinformatics, but also electronic medical records and electronic health records for how to do this better. Clinical usefulness, again, this is going to, as more of these tests are proven to be clinically useful, more and more of them are going to be used. We're still dealing with the roadblocks of payment uncertainty because payers do not fully understand genomic analysis, as well as regulatory uncertainty. So these are some of the issues in addition to the continued research, molecular research, that we also, I think, need to deal with in order for this to become realizable, a reality. Today, we really are at a tipping point. We are able to characterize disease at the molecular level more accurately, leading to increased efficiencies, better therapeutic responses, and reduced adverse events, and diagnostic and risk prediction processes. And all of that together is going to help us, has the potential to transform healthcare because it all makes a world of difference to patient care now and in the future. Thank you for your attention.
Thank you, Dr. Issa, for that excellent overview and really driving home the point that diagnostics are essential tools in understanding disease. And the more we understand about the complexity of disease, the more we can talk and deal with individual patients and what they're presenting clinically. We hope to have some time at the end of this session for a little bit of questions and some discussion. Does anyone who has an urgent question right now for Dr. Issa and doesn't want to forget it later? Okay, great. So let's move along then to Cesar Garcia and talking about one of his company's tests and some broader implications. Thank you.
Thank you, Andy. Good afternoon, everyone. I'm Cesar Garcia. I'm the Chairman and CEO of IRIS International. And probably you know our company more for diagnostics urinalysis and personalized diagnostics. IRIS is the market leader in urinalysis in the United States, so most likely every time that you go to your doctor and they perform a urine test, it's performed by one of our instruments. But today, I really want to talk to you about our personalized medicine initiative, and that hinges about a technology that we call NADIA, Nucleic Acid Detection Immunoassay, and our first application is for a prognostic prostate cancer marker. Moving on in terms of what is a prostate cancer problem. Prostate cancer is a very serious health issue. There's about 220 to 250,000 new patients discovered every year. People die from prostate cancer. The global incidence is about 700,000, so you combine that, it's about 1 million men suffer from prostate cancer every year. The treatment options are all significant. It's basically surgery, the radical prostatectomy or the removal of the prostate. It's external beam radiation therapy, radioactive sick implantation, or active surveillance, which doesn't mean to let the patient with no treatment. It means that the doctor decides to monitor the patient very closely until there is a reason to initiate treatment. 95,000 men every year go through a radical prostatectomy. And the number is increasing because of the early detection of prostate cancer, and also because the availability of minimal invasive robotic surgery, basically the Da Vinci robots that were commercialized by Intuitive Surgical. So a urologist decides to perform a radical prostatectomy when prostate cancer is confined to the prostate gland. And typically, what you do, you discover that by monitoring the prostate-specific antigen, performing a biopsy, or following the patient's symptoms. So let's say that the patient decided with the doctor to go for a prostatectomy. After the prostatectomy, the prostate is removed, and there is an anatomical evaluation, a pathological evaluation of the prostate tissue. So they look into the Gleason score. They look into the disease of the level, how big the tumor was, the spread of the disease, the pre-PSA level prior to the radical prostatectomy. They monitor the level of PSA, and based on that, they classify the patient between high risk, moderate risk, and low risk. To be candid with you, the issue of treating the low-risk patients is easy. Most of them are cured. Prostatectomy is very effective in cure prostate cancer, about 50% of the men are cured. The issue is the other 50% that are in the moderate to high-risk category. The doctors don't know what to do. The patients are very concerned because they were classified to be medium to high risk. And up to the release of Nadia Prosview this year, there was no diagnostic test to help them make that decision. There was an independent study performed in 1999 by Dr. Pound, and they took a cohort of patients that were not treated. These patients were classified to be low risk, moderate risk, and high risk. You can see the problem here. Of the 50% that were moderate, only 23% of them have biochemical relapse. Unfortunately, more than 50% of those patients, they receive today radiation therapy or androgen deprivation therapy, which is not needed. Even on the high-risk profile, 63% relapsed, 37% did not relapse. As you will see in the presentation, Nadia Prosview has a capability of helping doctors and patients to make that decision. We don't claim that Nadia Prosview is the only way to do it, but it's really a significant tool to make that decision. So what we did about 10 years ago, we began the development on an ultra-sensitive and very precise prostate cancer marker using the combination of two technologies: immunoassay to capture the PSA, which is a protein, the prostate-specific antigen, and real-time PCR to do the amplification. So Nadia combines immunoassay with nucleic acid, and that's what we call it, nucleic acid detection immunoassay. It basically provides an individual decision point for every patient by every doctor. For the patient, it's provide the clinical clarity for the patient at high risk. They don't know what to do with these patients when they're in moderate to high risk. Treatment of prostate cancer post-prostatectomy is a financial burden to the healthcare system. And also, one of the problems, big problems with prostate cancer treatment is not only the disease, it's lack of peace of mind. The man is very concerned because of the disease and the fear to die from prostate cancer. Now, what we did in Nadia, you basically is an ultra-sensitive protein detection system using the combination of immunoassay and real-time PCR. And in traditional prostate-specific antigen testing, you may get a value of 0 or close to 0. But 0 is not really 0, it's the limit of detection of the assay. So what we did, when you're counting in traditional immunoassay, you're counting probably to about 100,000 molecules. Nadia PSA can count down to single molecules. We also tested for the FDA, because it's really an assay cleared by the FDA. We tested the linearity and the precision of the assay. And if you see, the dynamic range of the assay is very high, very linear, so it's very sensitive and very precise. And I will explain to you why that is important. Nadia Prosview was cleared by the FDA in October of 2011. It's not a lab-developed test. It was developed with the rigor of any other diagnostic test. And the FDA required that to release Nadia Prosview, we have to confirm the recurrence of prostate cancer against a hard clinical outcome. So we're not confirming here that we're not trying to predict the elevation of PSA after prostatectomy. We're trying to identify that the patient truly relapsed by finding cancer in the imaging, in the biopsy results, or death of prostate cancer as a prostatectomy. How does Nadia work? Well, we take 3 samples, 3 blood samples, one 3 months after the prostatectomy, one 6 months, and one 9 months after the prostatectomy. We plot the 3 points, and we look into the slope of the 3 points. Now, very important, to be able to plot that line, you have to be extremely precise, because if you repeat the same patients and you get a cloud of points, you get different results, you cannot draw a line. To give an idea of what we discovered is that this man, stable men, they have a concentration down to about 2.5 picograms when the limit of detection of traditional immunoassay reliably is about 200 picograms. So we have to be about 100 times more sensitive than traditional PSA and very, very precise. So when we plotted 3 points, we basically developed a hypothesis, and the hypothesis was proved by the clinical study that we performed. The hypothesis was that a man that has an elevation, a rise of PSA of less than 2 picograms per milliliter per month, that patient is stable, and that patient does not need to follow treatment with radiation therapy or androgen deprivation therapy. If the patient is not stable, that patient must be followed. So the official claim that we got from the FDA was that Nadia Prosview is indicated for use as a prognostic marker in conjunction with clinical evaluation as an aid in identifying those patients at reduced risk for recurrence of prostate cancer for 8 years following the prostatectomy. That's a very strong claim supported by clinical data, and to our knowledge is the first time that the FDA approved a marker to diagnose stable patients after a prostatectomy. So our proposition is don't treat patients that don't need treatment. And that's a very important decision, it's a critical decision, and that's why the FDA took us through a very, very rigorous testing. The clinical study was very significant. We have to search for 27,000 patients. These are retained samples. Of the 27,000 patients that we sorted, we found about only 700 that qualify with all the criteria that the FDA required. We selected on a random basis about 300, and these are the results of that random study. Remember that this is not about predicting biochemical recurrence, it's actually confirming that prostate cancer was present in bone scans, in biopsy, or death from prostate cancer. The results are the following. We achieved 92% negative predictive value. So when you said that the patient is negative, 93% of the time we were correct. It's unusual to see a prostate cancer marker with a very high positive predictive value. Not only we achieved a 93% negative predictive value, we also achieved a 78% positive predictive value, which is very important for the doctors, because when you tell the doctor that patient is not stable, you're saying that that patient is positive. They don't want to be treating false positives. So the balance between the false negatives and the false positives is very important. Now, the FDA asked us to do something more. Try to do an analysis of those samples and correlate the slope to the actual recurrence. So we went back to the data, and we plot a Kaplan-Meier plot that basically plots the probability of being stable after a number of years of surgery. The power of Nadia Prosview is such that you see the green curve in there, 90-some% of the men were stable for 17 years. And 17 years was the limit of the samples that we collected. Probably were stable forever, but we don't know that. And we cannot make that claim because the data doesn't support it. But what is very telling is a separation between the two curves. When Nadia Prosview said that the patient is not stable, the probability of relapsing or having a clinical relapse drops tremendously in 5 years. The probability of being stable is only 50%. So when you compare the two groups and you compare the curve, basically the patients that were identified to be non-stable, practically all of them relapsed over the duration of the study. So the clinical benefits are many. It provides an objective tools that enable the clinical clarity to treat moderate and high-risk patients. It provides a tool for the doctors to optimize the management strategy for the men post-prostatectomy. Not only we want to optimize the clinical value, we want to optimize the clinical value by avoiding radiation therapy, androgen deprivation therapy, and to minimize the morbidity associated with prostate cancer treatment, which is typically sexual impotence or urinary incontinence. It provides peace of mind to the patients, and it provides also the potential that men that are cured, that they have an objective measure that they can go to their insurance company and say, I can be insured because I'm cancer-free. Today, they don't have a way to get that. And conversely, the men that are cured, but obviously they suffer significant sexual dysfunction and they need testosterone therapy. The doctors do not prescribe testosterone because it can increase the tumor again. But if they're cancer-free, they can take that therapy and resume a normal life. So approximately 25% of the patients post-radical prostatectomy receive radiation therapy or androgen deprivation therapy. It's very expensive. Radiation therapy costs about $50,000 per patient. That doesn't include the cost of treating the comorbidities that I mentioned before, urinary incontinence, sexual dysfunction, bladder and rectal cancer, proctitis. Androgen deprivation therapy, we come to about the same number because over 3 years is about 48,000 different type of complications. The cost of treating prostate cancer in the United States is about $2.5 billion a year. Our estimates are that if Nadia Prosview was adopted as a treatment paradigm, we could save about $1 billion to the healthcare system by avoiding unnecessary treatment. The path to getting this clear has been a long path. We began the discovery about 10 years ago with a patent from the University of Berkeley, California. We went through the FDA for a long process, about 3.5 years. The assay was cleared in September and was launched in March. And it's very important to mention this because what happens is we in the AdvaMed members companies, we go through a very rigorous development process. I can tell you now that there's already lab developed tests that are claiming to do the same, and they haven't gone through that. And many of them, what they're doing, they're claiming biochemical recurrence when the FDA clearly says that's not an indication of prostate cancer recurrence. Now, you have the 510K clear, you went through that rigorous process, it took you 10 years, $35 million were spent, we're not there yet. The second part is to get reimbursement. And right now, we're going to the market, the company's funding the utilization of the assay. The assay is expensive, it costs about $500 just to collect the samples. The price of the assay will be in the order of $3,000 to $3,500 per test. And with their first approach to get reimbursement, we're going to be addressing the integrated delivery networks, the Kaiser Permanente, Mayo Clinic, the Department of Defense, the Veteran Affairs Administration, and in parallel, we're working with CMS to get reimbursement. We require analytical papers, 2 of them are already published, clinical papers, 1 of them is published, the other one is in process, and we have a field experience trial, which is designed to prove the impact in patient treatment and the economic benefit of using Nadia ProsView to treat patients. Based on the actuarial analysis that we have performed, we believe that the savings to the healthcare system will be in the order of $10,000 to $15,000 per patient. If you avoid unnecessary treatment, that's why we price it at $3,500 so we can recover our investment and we can fund new technologies. So we expect to get reimbursement by 2013. Now, count when we began the process. It's going to take us 10 years to get through the discovery and the FDA process, another 2 to 3 years to get reimbursement. In that time frame, we could have been saving money. And this is really brings me to the conclusion of my presentation. There's a general idea that diagnostic tests increase healthcare costs. I don't believe that. I think that effective target therapies, effective target diagnostics, they have not only the potential of improving patient care, but they have significant potential to reduce healthcare costs. And like I said, in this case, $2.5 billion a year spent, we could save probably $1 billion. What we need improvements to the regulatory and reimbursement systems, better coordination between the agencies involved, so they can provide the support and expedite the implementation of this kind of innovation through the diagnostic industry. So that concludes my presentation. I can take any questions from you. Thank you.
Any pushback from the urologic community relative to the substantive samples that have to be taken, and quite possibly the patient community as well? And any thoughts about morbidity there? I assume you're going back to the OR to do the scoping and the procedures, correct?
Yes.
Any thoughts about morbidity there as well?
So the good news is that the test is a blood test. So we don't have to go back and check the patient. It's a blood test. The reception from the urologists have been excellent. We have many of the luminaries in prostate cancer are in our scientific advisory board. We began already testing in Johns Hopkins. We have talking to Kaiser Permanente, the Department of Defense. So the reception has been very good. Actually, one of the biggest concerns was the radiation oncologist, because we thought that we were going to basically get a pushback because obviously immense revenue for them. And the reality is radiation oncologists, they do not want to give radiation to a therapy that doesn't need it. And conversely, sometimes insurance companies do not approve radiation therapy when a patient needs it and they don't have an objective indicator to initiate radiation therapy. ProsView can help them in both ways. Yes. Thank you.
I noticed something really interesting in the beginning of your presentation,
Yes.
which is sort of related. I noticed you put a statistic about how many occurrences there are prostate cancer in the US and then around the world. And I noticed that in the US, that's a huge, you know, you have 228,000 out of the global incidence, that's almost a third.
Yes.
So in my mind, that probably spells some kind of environmental factor. Have you studied?
I think that it has to do more with the treatment paradigms. Remember that when you talk the total population, if you go into rural China and India, and you can't use those countries to count in the population because unfortunately, those people don't have the access that we have here. So, but the presence is very prevalent. There's about 1 million men that have survived over the last 5-6 years. So then 20 million men surviving prostate cancer, and that will be one of the follow-up applications to actually monitor those patients. We don't have that claim yet. Yes, sir.
Why CMS national coverage analysis rather than local carriers?
We're going to do both ways, okay? We're going to begin with Palmetto, okay, for California. But it's important to really address it in parallel. So the answer is we're going to go both ways.
No, CMS has a reputation for not liking when, CMS has a reputation for not being real fond of companies going parallel. I mean, are you not worried about a little bit of backlash from Louis Johnson?
We believe that we're going through Palmetto first, and then following with CMS will be the route. But one of the things that is important to keep them kind of informed of what we're doing, we believe that it's kind of opening the dialogue will be but the first priority will be Palmetto.
So you're going to Palmetto?
Yes.
Be the first,
Yeah.
and then
Then CMS, yes.
so it's not parallel.
Yeah. This is staggered.
Great, thank you, Cesar. I know
Thank you very much.
I won't even repeat the excellent points that Cesar summarized so well about the challenges and benefits of our industry as exemplified in that particular example. So let me turn it over next to Dr. Koch. Thank you.
Thanks very much. It's a pleasure to be in Washington. I spent quite a few years at 200 C Street with the FDA, just a couple of blocks from here, brought back some fond memories. I left in 1998 to join the molecular diagnostics industry. Roche Molecular Systems was formed when the PCR patents, polymerase chain reaction, were acquired from CETUS, and a whole new industry was started in molecular diagnostics. And I wanted to be part of that revolution, particularly this notion of personalized healthcare. The story I'll tell you about today is exactly that, marrying a diagnostic result to a therapeutic selection that has tremendous benefit for patients. So I won't spend a lot of time on this. I think you understand the general concept. Dr. Issa already told us that we've been moving towards a molecular-based definition of disease. And that's where diagnostics come in, to tell us that a tumor that sits in your lung is one of perhaps 20 different subtypes that has to be treated differently. And only with molecular diagnostics can you ascertain what type of disease you have and how to best treat it. Molecular diagnostics are leveraging some 30 years of scientific inquiry into a variety of diseases, but here we're talking about cancer. And sequencing of cancer genomes over the past decade is providing us a lot of granularity about these very diverse cancer types that are defined by molecular defects in the genome that are acquired over time. So if we can use a prism, such as a molecular test, then we're going to go from a situation that you heard described earlier of a generic chemotherapy, radiation therapy approach to one where, based on your molecular profile, you get a therapy that's going to be more efficacious for you with a better side effect profile and response. And that's precisely what we're doing. It's pretty obvious that leads to more effective drugs, it leads to fewer failures in the clinic, and also shorter development cycles to get new medicines to patients quicker. Who benefits? Everyone. Patients, payers, physicians, and drug developers. You can imagine, from the patient perspective, any better therapy when you've been told you have stage 4 cancer is going to be welcome news. The physicians clearly want to provide the best medicines they can for their patients, improve the efficacy, and reduce the adverse side effects. Clearly, payers want more value in terms of cost-effective health care. We as an industry want clinically differentiated medicines and tests. And last but not at all least, regulators such as the FDA and policymakers such as you in the room want to have improved healthcare cost and delivery of medicines. So it's a no-brainer. Why aren't we doing more of this? Well, there are a few challenges, and I'll share with you what some of them are and how we've tackled them. First, you have to find the right markers, so the science has to be really solid. You need to have early and significant investment in diagnostic development, and that's something certainly from the pharmaceutical development side that's been relatively foreign. This hasn't been part of their formula of drug development in the past. It changes how you do certain trial designs. Regulatory requirements vary widely. In the US, the FDA calls these high-risk PMA, pre-market approval type devices. In Europe, they're the lowest risk. You can self-certify such a test. The coordination has to occur between the two companies. And last, and also not least, there needs to be a clear incentive for diagnostic companies because, as we heard earlier, a laboratory-developed test can be introduced relatively easily without having to spend the millions that are involved in an FDA approval process and to clinically validate and prove the utility of that diagnostic in consort with the test, with the drug. So the story here is about melanoma. I don't know your experiences. I've met many people who have had melanoma. It's unfortunately increasing in frequency because we spend more time in the sun. UV light is what causes this. It damages the DNA. There are between 70 and 100,000 new cases in the U.S. every year, so about 1 in 50 people will get it. The good news is it's on your skin. You can see it. If you see it early, you can cut it off and you're going to do fine. But sadly, a significant number of patients, perhaps about 8,000 a year in the U.S., are diagnosed with metastatic disease. That means it's spread. And when that happens, it's a pretty dire diagnosis. In fact, until recently, about a year or so ago, all you had was dacarbazine, a cytotoxic chemotherapy that provided no survival benefit, but it was all we had. There have been some biologics introduced, but they have very low response rates, questionable survival benefit in some cases. They're difficult to manage from a toxicity profile. So clearly, there's a high unmet clinical need here for this disease. As a result of the sequencing of the human genome around the turn of the century, it's been 12 years now, scientists started to focus their attention on diseases to ask, are there specific variations in disease tissue, such as cancer, that is causing this disease? And what they found in 2002 at the Sanger Center is that there's a kinase, that's an enzyme that puts phosphate groups on other proteins, called BRAF, that is frequently mutated in a wide variety of cancers: melanoma, colon cancer, thyroid, and ovarian. And that had some very quick ramifications for drug development as well as for diagnostics. Here is a much more stylized and very simplified signaling pathway. You saw one earlier. Receptor tyrosine kinases sit on the surface of the cell. They receive signals from other cells, and they send it down this transduction pathway with a series of switches. Each one of those proteins, like RAS, RAF, and MEK, are essentially protein switches that tell the cell grow, replicate, or stop. What happens with BRAF is it gets mutated in the 600th amino acid from one amino acid to another, and that turns the protein on so it's constitutively signaling. The switch is permanently on. That's bad for cells because now they're proliferating continuously, and that leads to cancer. These are called driver mutations. There are hundreds of different driver mutations in the various kinds of cancer, but the good news is because it's a kinase, we know how to target those kinds of proteins. Our colleagues, Gideon Bullock and others at Plexicon, a startup biotech in the Bay Area, developed a molecule based on the structure of this mutated protein that is highly selective for the mutated BRAF protein. And so they knew that this would be important for a disease like melanoma, where 50% of patients have that mutation. And they approached us in 2005 and said, can you develop a test that identifies the presence of that mutation in cancer tissue? And of course, we said yes. A year later, as we had started that development, they filed an IND. Roche Pharmaceuticals became their partner to develop the drug. And early in 2009, the test was used to screen the first patients after dose escalation. For those of you not familiar with drug development, in phase 1, when you first introduce the drug into humans, you gently escalate the dose to a point where you, before you hit toxicity, and that's where you then start to look for efficacy. What was done here was after that dose escalation had occurred, About 35 melanoma patients were tested, and those that had the mutation, as identified by our test, were treated with the drug. And let me show you what happened. About 3 years ago at the ESMO ECHO meeting, these pictures were shown. These are FDG PET images, and you can see that before treatment, there's a huge mass in the cavity, center cavity of the patient, and that after only 2 weeks of treatment, that cancer had essentially melted away. These are not typical results with cancer therapies, and so you can imagine that the community, cancer community, was really excited about this possibility that 70% or so patients might respond to this. And here, just so you have another visual, a patient who put this up on his website, all the masses that are deposited under the skin, and after 4 months of treatment, they essentially were gone. So I think it gives you a sense of the power of directing therapies to the drivers of disease and why you want to develop medicines in this way. So these results were really excited, and everything kicked into high gear. Our pharma partners quickly initiated phase 2 and phase 3 trials, and as you can see, they're only about a quarter apart. That is atypical for drug development. Normally you do a phase 2, and after you've gotten those results, you invest in the much bigger phase 3 pivotal trial that's randomized. These were run concurrently because they were so confident the right patients were going to get this medicine and it was going to show benefit. The trials were run globally, over 2,000 patients screened within 1 year, and the outcome was that at the beginning of 2011, the results were unveiled for the BRIM-3 pivotal trial, and the data were filed with the FDA. They were filed end of April, and on August 17th of last year, the drug and the diagnostic were approved together. It is unheard of to see, first of all, a drug have an NDA filed and then approved within 5 years, and similarly, to have a diagnostic PMA approval in less than 4 months. But clearly, the FDA was highly motivated to get this medicine to patients who were going to benefit from it. But the other message here is that's a fairly long timeline from another perspective. If you think about the investment that went into both the drug and the diagnostic to gain this approval. The two package inserts reference each other. So in the Zelboraf drug label, it says that only patients who are detected with an FDA-approved test are recommended for treatment with this drug. And similarly, the drug is referenced in the package insert of the diagnostic. I will say that within 24 hours of this approval, there were a number of companies with laboratory-developed tests advertising that they had a test for Zelboraf patient selection. Clearly not what the FDA intends with the label here, and nor were those tests clinically validated in the trials the way that we did. Just to give you more granularity about that, you're welcome to look at the package insert if you like that sort of detail, but over 25 assay performance studies with 600 unique specimens to verify the analytical performance. Moreover, the clinical performance, and that's most important, over 2,000 metastatic melanoma patients were tested, and those who were positive were shown to benefit from this drug. A highly reproducible test across multiple labs, and importantly, the FDA regulates the entire system, the hardware, the software, the reagents, and they do so on an ongoing annual reporting basis so that you have a standardized test that works really well. A little conundrum we had, because technology is moving so fast, the FDA requires that the gold standard be used as a comparator for this. The gold standard for DNA variation is Sanger sequencing, and it's actually a bronze standard these days. It's really not as good as what you can do with PCR, and we had to use the next generation of sequencing to show that Sanger was wrong and that our test was correct. But the other reason I bring this up, there are laboratories who use that technology as a diagnostic today. And clearly, that would be a problem for patients that they could potentially be misdiagnosed. This is not yet routine by any means. There are many challenges, but the FDA is certainly working with us to try to make it more facile, both from the therapeutic and the drug and diagnostic development perspective.
I have one maybe for Dr. Issa. And could you give us maybe some specific sense of given all the, the advances to date, obviously the diagnostics industry in conjunction with the researchers are delivering a tremendous amount now. There's tremendous hope for the future, and there will be further advances. Can you give us a sense of where we might be in 5 to 10 years versus where we are now? Because we talked about this huge wealth of understanding and untapped amounts of genetic data that we're just starting to parse. Obviously, it's going to take a tremendous amount of time to come to a full understanding. But what in this sort of near midterm horizon can we expect to see actually being delivered?
It's always a little hard to get the crystal ball type questions, and you try to answer them at your own peril. I think the train has left the station. We're going to continue. The actual molecular research is going to continue, and it's going to continue at a fairly rapid pace. I think some of the successes we've seen over the last 6 months to the past year in particular have been quite exciting in melanoma, prostate cancer, lung cancer as well. And so because of that, I think we're going to try and continue, we're going to see more molecular diagnostic companies, and AdvaMedDx is certainly leading the way, trying to come up with the same model of companion diagnostics, targeted therapeutics, because that model works. Works for patients, it works as a business model, that sort of thing. But certain things have to happen in order for all of these, everything to be aligned. And the incentives have to be aligned. Regulatory aspects have to be aligned. So we need to have processes in place that make things even easier. The FDA is certainly motivated, and kudos to them. But I think there's still a need for getting through some of the roadblocks, both in terms of the payer community, the regulatory community, as well as other stakeholders.
Thank you. I want to take just a moment to recognize Vince Forlenza, who's been able to join us. He is chairman and CEO of Becton Dickinson, one of the global leaders in diagnostics. And one of the reasons I'm calling him out, not just to maybe embarrass him a little bit, is to say that he's also chairman of the board of the AdvaMedDx directors, and has exhibited tremendous leadership for this whole industry and with the formation and startup of this industry association. So really appreciate his efforts personally. I guess I would summarize simply by noting in a summary fashion what all of our speakers have indicated so well, that the diagnostics industry is delivering tremendous value right now to patients, is on the cutting edge of science and technology, and in the near term will be delivering even more value, value for the money and value for patients through advances in patient care and the diagnostic technologies that these companies are delivering. But there also remain challenges, and they're significant both in the scientific and policy arena. So I urge you to continue to educate yourself on this field. Use us as a resource. We're happy to point you in other directions as well. Really appreciate your attendance today, and we'd be happy to stay around and answer any other questions. Thank you.